Good morning, everyone. Can you hear me?
Yes.
Thank you. Hope you had a good weekend. It is Monday, November 23rd, the week of the Thanksgiving. So we'll have a lecture today, and then the next lecture will be a week from today, the last one. So what do we have? We have a signal transduction, biochemical signaling, a very important topic, right? So as we mentioned, cells cannot live by themselves. In a multicellular organism, they have to communicate with each other. And so that's what we are talking about, how they do communicate with each other and how they know what to do, right? So let me share my screen. Share my screen. Okay. So we discussed various types of receptors. And as I mentioned, GPCRs, G protein coupled receptor, right? G protein coupled receptor is one of the most common ones. And as we mentioned, 30% of all drugs exert their action through GPCRs, right? There are 800 different genes which code for GPCRs. And so epinephrine is one of the hormones which is made in adrenal glands, right? And that works through GPCRs. So epinephrine, as we know, it mediates stress response, right? Mobilization of energy. So it has a lot of receptors in muscle, liver cells, right? So basically diverting energy from organs that do not provide immediate response to flight, to support fight or flight response, such as stomach, for example, right? So the blood flow to the stomach is not necessarily important when you have to run away from something is going to eat you, right? What you do need, you need energy in your muscles. You need energy. You need to communicate to the liver, so the liver produces glucose and puts it into the blood, right? And also receptors in adipose cells, lipid hydrolysis, right? So you need to break down lipids again, so you produce energy. You need to have receptors in heart cells, right, which will increase the heart rate. And so there are lots of molecules that have been discovered. Now, this is the natural ligand, epinephrine, and this is its KD constant, right? Dissociation constant. Remember, the lower, the better, right? So epinephrine by themselves is 5 micromolar, and we can do better than that. So for example, isoproterenol, a drug, works in a similar way to epinephrine. And when we say agonist, do you guys know the difference between agonist and antagonist?
Yes, sir.
What's the difference?
Doesn't it increase the uh, binding and then antagonist will like block it kind of?
Yeah. So agonist basically, yeah, agonist binds in the same way as the natural ligand, right? And also induces produces the same outcome. Whereas antagonist just simply blocks the receptor. And in general, if I gave you, let's say, 10 different drugs with structures of agonists and antagonists, what you would notice is that antagonists are larger molecules. They're larger molecules, and specifically so that they actually block the receptor. They block the receptor, but because they're so bulky, they will not be able to produce the agonistic action. And you can see here that propranolol, which has been which is a beta blocker, has been in the clinic for a long time. People take it for a variety of conditions, to lower blood pressure, for example, and for anxiety as well. And you can see it's an extremely potent binder to the epinephrine receptor. An excellent drug for performance anxiety. Whenever you have to speak in public, for example, many people take propranolol. Okay. All right, so specifically, let's look in some detail how these G protein coupled receptors work. So epinephrine, or another ligand that binds to GPCR, there's a binding pocket here, right? So this is beta adrenergic receptor, so epinephrine receptor. Remember epinephrine and adrenaline are just two different terms for the same ligand. And so G protein is a heterotrimeric protein. It has alpha, beta, and gamma subunits. And as the receptor binds to the G protein, it binds on the alpha site. On the alpha site, and the GDP, guanosine diphosphate, undergoes exchange with GGP. So GGP leaves, GGP comes in, right? Remember, the difference is this extra phosphate group on GGP. So once that has happened, the beta and gamma subunits dissociate, and the alpha subunit is now all by itself and is capable of binding and activating an enzyme known as adenylyl cyclase. And the function of this enzyme is to take ATP, adenosine triphosphate, and convert it to cyclic AMP. We're going to look at the structure on the next slide, but for now it's just cyclic AMP. Okay, so it's a cyclic phosphate, and so this one, the role of this one, oftentimes this is referred to as second messenger. So we'll talk about some second messengers later. I'll give you more examples of second messengers. It kind of makes sense, right? So if your epinephrine is the first messenger that comes into the cell, that gives rise to another small molecule, which now is a second messenger. So this cyclic AMP then activates an enzyme, protein kinase A, pKa, which is capable of phosphorylating various cellular proteins and induce cellular response to epinephrine. And so cyclic AMP signal can be shut down, right? So there is an enzyme known as cyclic nucleotide phosphodiesterase, which will come in, break down cyclic AMP to 5' adenosine monophosphate, which will reverse the activation of pKa, right? So this is the way the signal is shut off. So this is a cartoon-like representation of how GPCR signal transduction works. And so here's our second messenger, cyclic AMP. And you can see the way it's produced is through this enzyme, adenylyl cyclase. Basically, it will break the diphosphate unit, and the 3' hydroxyl will form a phosphodiester with the 5' terminal phosphate, right? So 5' and 3'. So adenosine 3', 5' cyclic cyclic monophosphate, right? So there's only one phosphate. So it's a monophosphate as opposed to triphosphate, but it's also cyclic. And then, so this molecule by itself will then can be cleaved, right? It's actually quite a strained molecule, so it can undergo hydrolysis right here. So the water will come in, attack the monophosphate, and this will be the leaving group. This will be the leaving group to generate adenosine 5' monophosphate. And in the cell, then this undergoes phosphorylation. Various enzymes known as kinases, right, will then turn AMP into ATP. And the cycle basically is ready for another round of pKa activation. All right. So pKa, so cyclic AMP allosteric is an allosteric activator, right? So it binds at a site different from the site where the protein kinase A phosphorylates various proteins. And it also leads to activation of enzymes that release glucose from glycogen, right? So you need a lot of glucose in the bloodstream if you need to run for a long time. And so we talked about the fact that many receptors provide what's known as a signal amplification, right? So just look at that, for example. So just a few of GPCRs, right, will lead to the activation of a number of adenyl cyclase enzymes. Every adenyl cyclase enzyme will make several cyclic AMP, thus activated several pKa enzymes. These will activate thousands of glycogen degrading enzymes in the liver tissue, and tens of thousands of glucose molecules are released in the bloodstream. So here is this a very rough approximation of how this works. So epinephrine, you just need one molecule of epinephrine to basically bind to the G protein, right? G protein will then dissociate, producing the alpha subunit. And this can happen with just epinephrine. This can happen with, so epinephrine does his job here, then it jumps over, does a similar job at a different G protein, and so on and so forth. And so 10 molecules of G alpha subunit will be produced. If so each of these 10 molecules of G alpha subunits will then activate one molecule of adenyl cyclase, and this enzyme will then produce 200 molecules of cyclic AMP from one molecule. Sorry, from 200 ATPs, it'll produce 200 cyclic AMPs. Then cyclic AMP, each molecule can activate 100 molecules of pKa, protein kinase A. Then each molecule of protein kinase A can phosphorylate 1,000 molecules of phosphorylase B and convert that into active phosphorylase B kinase. Inactive glycogen phosphorylase B, another 10,000 molecules. And then glycogen will be degraded and 100,000 molecules of glucose, 1-phosphate, will be released. And once the phosphate is removed, the glucose will enter the bloodstream. So you can actually multiply all of these and it'll give you specifically how many molecules of glucose will be produced from one molecule of epinephrine. So very important phenomenon, very important characteristic of such a G protein coupled receptor is signal amplification.
Dr. Kornienko?
Yeah.
I know there's like GI that inhibits the G protein. Would that just inhibit the formation of the cyclic AMP?
GI. What is GI?
It inhibits.
Inhibitory?
Yeah, it's inhibitor.
Well, we'll talk about different. No, I don't know in the answer to your question.
Okay.
I don't know the answer to your question. GI is. I think I've seen it somewhere, but I don't know much about it. So. GI, does it form when you phosphorylate the tyrosine residues on the G alpha? Is that what you're talking about?
I think it's just like a generic term for something that inhibits the G protein coupled receptor.
Okay, let's get to that. I think in the next couple of slides, I'll tell you how it's desensitized, and maybe there we can discuss what you just asked. So. Right, so cyclic AMP, you can see is, as I mentioned to you, biology is a very biological systems are very conservative, right? They don't want a separate molecule for every single pathway in the body. They utilize similar pathways, but just tweak them around, modify them a little bit, and use them in more than one signal transduction pathways. So you can see here, so some enzymes and other proteins regulated by cyclic AMP, dependent phosphorylation by pKa. And here's a list of those. And I think so this S, you can see in red, right? So that's where phosphorylation occurs. So pKa is phosphorylates other enzymes on a serine residue, right? Remember, serine is an OH. So it's not a tyrosine kinase, it's a serine kinase enzyme. Serine or threonine. You can see here also there's a threonine that can be phosphorylated, for example, on protein phosphatase 1 inhibitor 1. Okay. So as far as deactivation of this signal, so let's talk about that. So epinephrine in general is a short-acting signal, right? So you want to mobilize energy, but you don't mobilize the energy for hours. You don't, at some point you would like your food to be digested. And so it's a short-acting signal. So so basically you run for a short period of time, run away from the predator, and then you want obviously the concentration of glucose in the blood to be normalized, all right? And so downregulation of cyclic AMP then occurs by hydrolysis of GTP, right? So the receptor is also known as a GTPase, which means it's a receptor that can shut itself off by hydrolyzing GTP in the alpha subunit. And we'll talk about desensitization or adaptation. So you can see here, this is the basic representation of how this will work. The signal activates the receptor response, and at some point when the response is no longer needed, an inhibitory molecule will bind allosterically, usually to the receptor, and force some kind of conformational change. And so the signal can no longer be transmitted through this receptor. So So the self-activation, so for basically, again, once the first 3 steps have occurred, just like what we talked about, right? The G protein, GDP replaced by this ligand binds, GDP gets replaced by GTP, right? And then at some point, this GTP, which makes the alpha subunit active and activates adenylyl cyclase, then eventually GTP will be replaced by GDP. This no longer capable of activating the adenylyl cyclase. It comes back and reattaches to beta and gamma subunits and reattaches back to the receptor and makes the receptor and goes back to the original form, right? Waiting for the next molecule of epinephrine. And so the key here is the hydrolysis, right? So you actually need a molecule of water. So GTP is hydrolyzed to GDP, and that receptor actually accomplishes by itself. Okay. So as far as desensitization, so this is how this works. So so one way is obviously to shut off the signal. So for example, there's too much epinephrine, just like with any ligand, if there's too much ligand, like for example, you know that with many drugs that not only those that occur through GPC, whose action occurs through GPCRs, other receptors are also very similar. They can undergo desensitization. And so the best way to observe that would be, let's say, if you take a medication, right, and you need a certain dose. You take the medication, it helps you, but then after a month or 2, it stops helping you. So you need to increase the dose. And then another couple of months, increase the dose again. So what's happening is desensitization of the receptor, right? So the receptor basically is telling you that you need more and more of the drug to activate me, right? So the biology is undergoing adaptation to your drug. So so here is our epinephrine bound to the receptor. Dissociation. Okay, so then G protein undergoes dissociation to give you the G alpha, right? So this is so G alpha is not shown. We're just going to be looking at beta and gamma, right? So what happens to beta and gamma once the G alpha has departed? Beta and gamma, they still stay associated with the receptor itself, right? And then that complex of the receptor and the beta and gamma subunits of the G protein, they recruit another enzyme known as beta adrenaline receptor kinase, adrenaline receptor kinase, ARK. And so what this does, this will phosphorylate serine residues on the receptor. So there'll be 2 serine residues, and now you can see the serine phosphates. These 2 serine residues, now they obviously each of these has 2 negative charges. This will recruit another protein from the cell known as beta-arrestin. Beta-arrestin, which will bind to the phosphorylated residues on the receptor. And once that happens, the receptor beta arrestin complex will enter the cell, will basically come off the cell membrane and will go inside the cell and undergo endocytosis. Remember I told you endocytosis is when something comes into the cell as opposed to exocytosis, right? Exocytosis. And so there will be these endocytic vesicles where the receptor, instead of being degraded, right, and it's going to be stored, right? So once you already made the receptor, it's going to be a huge waste of energy to break it down and generate a new receptor later. So it's much wiser, right, to keep the receptor inside the cell so that it can be recycled when its activity is required again. So it will be stored in this endocytic vesicle. Arrestin dissociates, receptor is dephosphorylated and returned to the surface when it's needed again, right? So this is known as desensitization of the beta adrenergic receptor. So as far as GI, yeah, I don't know. We need to look this up. So you said so GS is the stimulatory, right? So what we've been discussing so far is the stimulatory G protein coupled receptor. Now, as far as inhibitory. What's the difference between GI and GS? G protein can be classified in 3 categories: GQ, GI, GS. Yeah, you see, this is beyond my knowledge as well. GQ and GS are stimulatory, whereas GI is inhibitory. GS activates cyclic AMP but they don't say what GI does.
It's just the inhibitory. It's up there. And then GQ just activates phospholipase C, which cleaves phospholipids.
Yeah, PLC. Yeah, we'll talk about PLC in a moment. But GI inhibitory. But what's the pathway for GI? Which enzyme does it activate or inactivate at the cell membrane? Anyway, we can look this up later and maybe chat about this later. Anyway, again, the purpose of this class, guys, is for us to, the detail is, obviously there's much more detail, right? To any of these, that just imagine if 30% of all the drugs work on GPCRs, you can imagine how much knowledge has been acquired through this, right? So many pharma companies are involved in working with GPCRs, so we know a lot about them. So so maybe this one, inhibiting, inhibiting and activating inhibiting cyclic AMP synthesis. Okay, human genome encodes for 1000. I give you 800 number. Maybe my data is a little outdated, but it's close to 1000. Hormones, growth factors, neurotransmitters, taste and smell. And as I mentioned to you, many of them are orphan receptors, right? Anybody remembers what an orphan receptor is?
A receptor with unknown function, or what binds to it.
Right. Specifically, the ligand, the endogenous ligand is not known, so we don't know exactly what it does. All right, so so cyclic AMP, some signals we use cyclic AMP as second messengers. All shown here. You can see epinephrine is one of them, but there are many others. There's dopamine, glucagon, various neurotransmitters, various physiologically important processes utilize cyclic AMP as the second messenger. All right, and so obviously there are a lot of opportunistic and a lot of organisms which utilize GPCRs for their own function, right? For their own survival. And so, for example, here, there are some bacterial toxins which will utilize NAD+. So for example, this one is cholera toxin and pertussis toxin, right? So the way they will work is there is arginine. There's an arginine group on the G alpha portion of the GPCR. And normally, remember, it's a GTPase activity which will shut off the signal. But what these two bacteria will do, they will basically use NAD+, and they will make this nitrogen attack the alpha position of the ribose. This will be the leaving group. It's a good leaving group because nitrogen is positively charged. And now you have this ADP ribose attached to the arginine moiety of the G alpha subunit of the GPCR, right? And in this state, when you have this ADP ribose attached to it, it loses the GTPase activity. Right, so the signal is no longer shut off. And so what will happen, obviously, then too much of cyclic AMP will be produced from ATP, and the signal will be constitutively active. Right, so even though there is no epinephrine anymore, but we're gonna be experiencing the effects of as if there was so much epinephrine. So in other words, we will continue experiencing the effect of activation of GPCR, right? Even though there is no epinephrine. All right, so secondary messengers can activate other secondary messengers. So this is a very interesting cartoon slide. So so this is, we talked about PLC a little bit, phospholipase C, another very important enzyme that is involved in the cleavage of lipids. And so remember lipase, remember I told you lipase is like esterase, only cleaves esters associated with lipids. And so let's see what happens here. So this is another signal transduction pathway, which is GPCR based. So in this case, we have some kind of hormone that binds to a specific receptor. So here is the receptor. This is this hormone. This is the heterotrimeric G protein coupled receptor with GDP bound to it. And so the occupied receptor in step 2 causes the substitution of GDP by GTP. So GDP leaves, guanosine triphosphate comes in, and obviously the beta and gamma subunits dissociate, right? And so in this case, it's actually GQ. Oh, that's interesting. GQ alpha. GQ alpha, it's also stimulatory, right? So stimulatory alpha subunit that binds to the enzyme PLC, phospholipase C. Phospholipase C, which cleaves the lipid phosphoinositol bisphosphate, PIP2, and it will cleave it in such a way that there will be a second messenger generated, inositol triphosphate, which is known as IP3, inositol triphosphate. And once the inositol triphosphate is cleaved, what's left, we have a lipid without the head group still stuck there in the membrane. And so that will be diacylglycerol, just diacylglycerol, right? So it's a molecule that looks like this. So we just cleaved off the phosphate. So you have one fatty acid, another fatty acid, R prime, right? And the OH group. So that's where the phosphate with the inositol was, right? And so that's gone now. And so inositol triphosphate then travels, now it's water soluble, right? So it's inositol plus 3 phosphates, highly charged, travels to the endoplasmic reticulum and opens the ion channel, specifically potassium channels, right? Potassium channels, they open, and one function of endoplasmic reticulum is a place where calcium is stored in a cell. And so inositol triphosphate will open the calcium channels. The calcium will come out following its concentration gradient, right? In most cases, the calcium concentrations in cells are kept low, and that's specifically for that reason. So when calcium comes in, that activates the cell to do something. And so you can see here, so calcium comes in, travels, and activates protein kinase C together with diacylglycerol. So it's like 2 second messengers. One travels through the membrane, so it's a lateral diffusion, and the other travels across the cytoplasm, and both bind to protein kinase C, PKC. Protein kinase C, and which is now attached to the plasma membrane. So by itself, it doesn't really have any hydrophobic moieties for it to attach to the membrane, but through diacylglycerol, it will attach to the membrane. Okay, and now once it's attached to the membrane, it can perform lots of phosphorylation reactions. So phosphorylation reactions, and then once it starts phosphorylating other proteins, the signal will be transduced further. Okay, so so this is just an example of how 1 second messenger can, which is, for example, IP3, right, can increase the production of another second messenger, which is calcium or diacylglycerol. And so these are small molecules. All right, any questions? Okay, so as far as calcium, so as I mentioned to you, calcium in the cell is a rare ion, but when it is there, that means that something is happening. Something is happening, and one of the sensing proteins is calmodulin. Calmodulin, if you talk to biochemists, they will tell you that lots of things occur through calmodulin, and calmodulin is a calcium binder. So you can see here there is a central alpha helix, in calmodulin, and there are 4 calcium binding regions. And so what happens, calmodulin usually binds to all other proteins through the central cavity. So this central alpha helix bends and surrounds the calmodulin regulated protein. And that is only possible if there is calcium, right? If there's no calcium bound to calmodulin, this will not happen. And one thing to remember, because this may be on the exam, just a random question. So you can see here there is a there's this EF arm, EF hand, right? Shown here. That's actually where there's this loop region and the alpha helix come together and form a nice binding site for calcium. Form a nice binding site for calcium. So there are 4 of those in calmodulin. So calmodulin is commonly abbreviated as CAM. Calmodulin. And so you can see here, calmodulin is ubiquitous, and there are lots of different enzymes and proteins which are regulated by calmodulin. Okay. Okay, so this is a little busy. This slide is a bit busy, but let's get through this. So this is how our vision works. How our vision works. You're looking at the screen, and that's what's happening in our rod and cone cells, right? Of the light sensing neurons. So so what's happening here is so we have a protein known as opsin, which can bind a lipid known as 11-cis retinal. 11-cis retinal, and when the protein opsin binds the lipid, it becomes rhodopsin. So rhodopsin is now a complex of a lipid with the opsin protein, which is ready for the to be activated by light. So light then hits the 11-cis double bond. And remember, I was telling you that whenever you have cis double bond, oftentimes you need some extra energy to go to trans. Cis, trans. So when cis undergoes isomerization to trans, basically rhodopsin changes its conformation. And so it's also, there's also a sort of G protein coupled protein, only here it's abbreviated as T, known as transducin. Transducin. T is transducin. Transducin. Again, the alpha, beta, and gamma dissociate. Alpha transducin then activates the enzyme known as phosphodiesterase. Phosphodiesterase that removes this inhibitor, and so the alpha subunit removes the inhibitor from the PDE, from phosphodiesterase, activating phosphodiesterase. And remember what these phosphodiesterase enzymes do. They take cyclic nucleotide and convert it to linear nucleotide, right? So cyclic GMP goes to GMP monophosphate, 5' monophosphate. And what happens here is that cyclic GMP originally, it kept this channel open. It kept this channel open. This is a non-selective channel, which would allow sodium and calcium to come in, right? And once this cyclic GMP is removed, there's no longer ligand that would keep the channel open. And so this channel will close. The channel will close from here to there, which means that the calcium sodium pump was sodium antiporter shown here. You can see there are one calcium going out, 2 sodiums are coming in, right? So if you close this channel up here, because cyclic GMP is no longer present, this antiporter will still continue taking calcium outside of the cell. Then the concentration of calcium in the cell drops. Calcium drops. And as that happens, reduction of calcium activates guanylyl cyclase. So this leads to, once this happens, this leads to hyperpolarization of the membrane. Remember, membrane is polarized, right? So the outside of the membrane has a positive charge. The inside of the membrane has a negative charge. And so obviously, if you continue taking calcium ion, if you close this channel, not allowing the positively charged ions to come in, right, you will cause hyperpolarization of the membrane. And the signal will be transmitted to the brain, right? Will be transmitted into the optical nerve, and the optical nerve will integrate these signals from various neurons, and specific nerve signal will be produced. So, so the rest of this is restoration, preparation for the next photon. So in the absence, so once the calcium concentration drops, guanylyl cyclase will make more of the cyclic GMP. Rhodopsin here, which was phosphorylated, right, which was phosphorylated, kind of bleached, will be dephosphorylated, so arrestin will be removed. Arrestin will be removed. Rhodopsin will be released. All trans retinal will come off. 11-cis retinal will come in, and this will be ready for the next photon. So whatever you see on top is the reception of the photon. What you see on the right is the hyperpolarization and production of the nerve signal. What you see on the bottom is the preparation of the system for the next photon to arrive. So this is how GPCRs work in vision, right? So here you can see there's no ligand, no growth hormone. It's actually light, which is the molecule that activates the G protein coupler receptor. All right, so let's see where we are. We still have to talk about enzymes as receptors. We'll talk about insulin in quite some detail. And that's probably the major concept that we need to discuss, how insulin works, and maybe sodium, potassium, and TPase. And we'll look at the ion channel and nerve signaling. I think that should be sufficient for next Monday. So the homework is open. You can start looking at it. Did you guys take the extra credit test? How was that?
It was good.
Yeah, any questions about that? Yeah, so as I mentioned to you in my message, I realized that for some reason, you know, I played with that, with the software, but I couldn't figure out how how to turn that into extra credit. So what I'll do, I'll just create a separate column and manually enter your grades as extra credit. So what you see right now for your final grade is your final grade is going to be higher than what you see right now. So I will take care of that today or the first thing tomorrow morning. Sounds good. Any other questions?
I think you can do the extra credit the same way you did the test 1 point adjustment. It's the same extra credit. It's just the same way on Canvas.
Yeah, you know, there is a way I can assign you quizzes. I can make two kinds of assignments, just assignments or quizzes, right? So if I just give you an assignment, I can, when it asks me out of how many points, I just put 0 there, and then it becomes extra credit. With quizzes, for some reason, it doesn't give me that option. And I Googled yesterday to see if anybody else has has encountered that problem, but I couldn't find anything. So I don't know. It's my first semester using Canvas, but the most important thing is that at the end of this class, we all, you know, you guys gain the knowledge that without any kind of technological, without having technology impact your grade, right? So that's my goal. So far, it's been good. All right. Anything else? Okay. Have a good Thanksgiving. I'll see you
Thank you.
on Monday. Bye.